Okay, let's move on to the next part.
Sure. So certain cells can certainly transform cholesterol into reproductive hormones or adrenocortical hormones. Certain cells, hepatocytes, can transform cholesterol into a bile acid. I don't think there's any other cell that can change cholesterol into anything else. So when people talk about cholesterol metabolism, there is no cholesterol metabolism. It can be converted into something in specific tissues, but it can be excreted. That's it. There's no.
All right. So, what I want to say here is that when I listened to the interview, again, the scope was limited to functions of cholesterol with respect to cell membranes, organelle membranes, and steroids, end products of cholesterol metabolism, right? So the steroid hormones like the sex hormones, glucocorticoids, and so forth. They really didn't talk at all about a very, very important distinct function of cholesterol, which is immune regulation. Completely missed, right? But it changes everything. And I think it's highly important that they consider this part, because if you're just looking at structure of cell membranes and steroids, then you're missing a much bigger picture. So here is something that I discussed on episodes 46 and 50. And we're looking at immune function of cholesterol. Here I focus on the innate cells, macrophages. Yes, yes, I love the macrophages, but we do also have this impacting adaptive cells. But just for now, let's look at this. Macrophages rapidly upregulate cholesterol synthesis when activated. And it turns out interferon signaling generate interferon responses that downregulate cholesterol biosynthesis and increase cholesterol storage in the form of cholesterol esters. Macrophages shift their cholesterol metabolism in a context-specific manner to facilitate specific effector functions and ensure the generation of particular forms of inflammation, interferons versus interleukin 1 beta, for example. So what we're seeing here is that by shifting their cholesterol metabolism, we are generating different cytokine signaling, right, to bring on specific forms of inflammation. It's very important in the immune function of cholesterol. The other thing that I think is missing in their consideration is the role of metabolites in biological functions, right? So again, here I show you the pathways for cholesterol synthesis, and they focused on the endpoints, the steroid hormones, the bile acids. They didn't talk about the oxysterols. I do have an episode on that. You can look that up. And then I think it was just an oversight. They forgot about vitamin D. I'm sure they're well aware. You all know it's derived from the synthetic route to cholesterol synthesis, dependent on the last enzyme in the Candet-Russell pathway. Okay, but they're focused on these endpoints. What they're actually not looking at are the metabolites, right? So protein prenylation, geranyl pyrophosphate, and farnesyl pyrophosphate, they are products of the cholesterol pathway, HMG-CoA reductase pathway. This means that statins, which inhibit HMG-CoA reductase, inhibit the production of both cholesterol and these isoprenoids, right? These are called isoprenoids. So, what's so important about these isoprenoids? Well, this is from a paper from 2007, and it says post-translational prenylation of proteins with isoprenoid lipids is one of the most widespread and well-recognized post-translational modification of proteins in eukaryotic cells. So, in this study from ACS Chemical Biology, in normal healthy cells, the function of the RAS superfamily of GTPases in diverse cellular processes such as growth, cell movement, protein trafficking, critically depends on their presence in the correct cellular membrane. And here we have a diagram from that study, and it's called Protein Prenylation, Enzymes, Therapeutics, and Biotechnology Applications, right, from 2015. And they basically tell you how, we sort proteins in the cell membrane. When we attach these isoprenoids, the farnesyl and the geranyl isoprenoids, to the protein, it helps their localization to specific parts of the cell membrane, right? And this also will help define polarity in the cell membrane. So this is highly, highly important for functioning, for signaling. So when people talk about cholesterol metabolism, there is no cholesterol metabolism. It can be converted into something in specific tissues. And by this, they're talking specifically about the steroid hormones mostly, right? And it can be excreted, and that's it. So it's completely missed out also the metabolites. Now, here's a reason, concrete reason, as to why this is important. Let's take a look at this. A fundamental role of pancreatic beta cells to maintain proper blood glucose level is controlled by the RAS superfamily of small GTPases that undergo post-translational modifications, including granulation. This covalent attachment with either a farnesyl or a geranyl group controls their localization, activity, and protein-protein interactions. Most small GTPases involved in insulin trafficking and secretion are required to be prenylated to function for their biological role and interaction with their respective effectors. So again, you'll see that these GTPases, they're not going to work and, then help with insulin trafficking and secretion if they are not prenylated. So again, people talk about studies where, there seems to be some correlation between use of statins and diabetes. Oh, that's no big deal. That's probably not important. Yes, it is. No, it isn't. I don't, I really focus on mechanism. For me, I like to know what might be going on. Again, I'm not saying this is the reason there might be an increased incidence of diabetes with statin use, but it could be one mechanistic pathway for that to happen, right? If we're not prenylating these GTPases correctly, then we might actually be interrupting insulin trafficking and secretion. Oh, and here, another study, but this one's talking about mTOR, right? Rep-G proteins and the activation of mTOR. I actually talk about this in episode 21. I do talk about mTOR and cholesterol and how that's related. That was a whole episode on mTOR, so if you want to take a listen, go ahead. But remember the small GTPases that are important in mTOR regulation and activation, right? And if they're not prenylated properly and they don't work, then your mTOR is going to work either. So again, might this be related mechanistically to statins' effects on muscle? That's something to consider. I'm always much more interested when there's a mechanism that's proposed. All right, my clarifications here then. Besides cholesterol, the mevalonate pathway is important for the synthesis of steroid hormones, oxysterols, isoprenoids. The metabolites of the pathway regulate cell function and metabolism in fundamental ways. Halting the mevalonate pathway not only means decreasing the end products of the pathway, it also decreases the metabolites of the pathway, and decreased metabolites of the mevalonate pathway impact cell regulation, proliferation and growth, signaling, metabolism, immune function, beta cell function,